In the following, we will discuss the two main applications of infrared spectroscopy in the life sciences. These are the analysis of the secondary structure of proteins and the analysis of the molecular reaction mechanism of proteins. We will start with secondary structure analysis, which is the most common application of infrared spectroscopy in the life sciences. Secondary structure analysis is mostly done using the absorption of the amide-1 vibrations. These are vibrations of the amide groups in the protein backbone. They involve mainly the CO stretching vibrations. Further contributions come from the CN stretching vibration and the NH bending vibration. All these vibrations are coupled in the amide-1 normal modes. There are as many amide-1 normal modes as there are amide groups in the protein backbone. The absorption of the amide-1 vibration generates an absorption band in the absorbance spectrum of protein, which is called amide-1 band. It is located in the spectral region from 1700 reciprocal centimeters to 1600 reciprocal centimeters, which is also called the amide-1 region. But this does not mean that only the amide-1 vibrations absorb in this spectral region. Side chains and polynucleotides, for example, absorb also in the amide-1 range. Nevertheless, in typical spectra of biological cells or tissue, most of the absorption in the amide-1 region stems from the amide-1 vibrations. A practical problem is that water absorbs also in the amide-1 range. Water absorption is usually stronger than protein absorption. It has therefore to be subtracted from the sample absorption or to be avoided by drying the sample or by using D2O. D2O absorbs at lower wave numbers outside the amide-1 range because the deuterium atoms are heavier than the hydrogen atoms. This is an example for the influence of the mass on the vibrational frequencies. This video shows the amide-1 vibration of a single amide group. As you can see, the stretching vibration of the CO bond has a large amplitude. When the CO bond extends, the CN bond contracts. In addition, we have two bending vibrations, those of the NH and of the C-alpha H bond. This figure shows the infrared absorbance spectra of two proteins in the amide-1 range. The red spectrum is the spectrum of albumin, which is predominantly alpha helical. The blue spectrum is that of avidin, which has a high content of beta sheet structure. Obviously, the spectra of the two proteins are quite different, and this is generally the case for alpha helical and beta sheet proteins. Many studies have correlated specific secondary structures with absorption in specific spectral regions within the amide-1 range. These regions are listed in the shown table, but there is no need to remember these numbers. Important is that beta sheets and alpha helices can be distinguished because the main beta sheet band is found at lower wave numbers than the alpha helical band. This is the main beta sheet band, and this is the alpha helical band. The alpha helix band overlaps with that of irregular or random structures when the experiments are done in ordinary water, H2O. Here and here. In D2O, the overlap is less. Here and here. These empirical correlations have been confirmed by statistical analysis of the spectra of proteins with known structure. These can be used to obtain spectra of pure secondary structures, as shown here. These spectra confirm the empirical correlations, but they do not make the distinction between turn and irregular structures. The main beta sheet absorption here is at lower wave numbers than the absorption of helices and other structures. The beta sheet spectrum has also a high wave number shoulder, shown here, that is often associated with antiparallel beta sheets. Note again that the absorption of alpha helices is found in the same spectral region as the absorption of other structures. However, the helix band is narrower due to a better defined structure. This exemplifies that the bandwidth reflects the conformational heterogeneity of the absorbing structures. We turn now to the reason for the structural sensitivity of the Amide 1 band. Its shape and position is determined by two main influences. The first one is hydrogen bonding to the CO oxygen and to the NH hydrogen. Both hydrogen bonds decrease the wave number and the frequency of the Amide 1 vibration. And this is an example for the sensitivity of vibrational frequencies to interactions with the environment. The second reason is that the Amide 1 vibration of a particular amide group is coupled to the vibrations of several other amide groups. The consequence of the coupling is that the Amide 1 vibrations are collective vibrations. This means that several amide groups participate in each Amide 1 normal mode. Another consequence is that the frequency of the Amide 1 vibration depends on the structure of the protein backbone. And this is an example for the structural sensitivity of coupled vibrations. This video shows one Amide 1 normal mode of a parallel beta sheet. You can appreciate that several amide groups vibrate at the same time and with the same frequency. The CO vibrations go through their equilibrium positions at the same time, as required for a normal mode. You can also see that some groups vibrate a lot while others participate less. For example, in the bottom strand, the second and fourth amide groups have a large vibrational amplitude, but those at the end of the strand vibrate only little. Amide groups which are neighbors in the chain vibrate out of phase in this particular normal mode. This means that one contracts when the other expands and vice versa. Summarizing what was said so far, the absorption in the Amide 1 range of the infrared spectrum is used to analyze the secondary structure of proteins. Two influences are important for this structural sensitivity: hydrogen bonding and the coupling of Amide 1 vibrations of several amide groups. These influences determine the band position of the Amide 1 band and make it sensitive to secondary structure. But not only to secondary structure, as we will see at the end of this section.